So thanks for the invitation. So today I'm going to talk to you about explainable AI for deciphering biological rules. So I will share with you my experience on that. So the context is the following. So we can observe several traits in humans. So the traits are as diverse as the color of the eyes or as the presence or an absence of a disease. And these traits are emerging from multiple factors, including biological factors such as genome, the epigenome, the transcriptome, the proteome, the metabolome. And we would like to study how a phenotype emerged from this set of data. So we can see this kind of question as a classification problem. So we have a set of phenotypes, and we want to classify the phenotypes from the omics data. So the pipeline is really standard. So we need to gather exploitable and standardized public data set with multi-omics data. So this is, so for images, there are a lot of data set available, and for biology, there is also a lot of data, but they are not shaped in a way that they are directly exploitable by machine learning researcher. So this is the first step. Then we need to define a task and to learn to solve it with an algorithm. And then we need to explain the decision of the algorithm. And so my slide is okay. So and so to explain the kind of explanation we can have would be what features the algorithm is relying on to do its prediction. So for instance, what genes it is using or things like that. So for this talk, I will use an attribution-based XI method. So we already had a lot of examples of such method for images. So this is just another example. So this is just to say that the explanation method will output for an image, it will output the pixels that are important for the algorithm for its prediction. And so we want to use this kind of method on our biological data and see if we can have some nice interpretations. So in our case, so we tried many experiments on one data set, which is a PanCan TCGA data set. So it contains many samples of cancer cells. For the algorithm, we just use a logistic regression because the task is pretty easy and we do not need to train a neural network. And then for the attribution-based method, we used integrated gradient. So for the data set, so in more detail, so we have 33 classes, we have a lot of samples, and we have a lot of genes. So the genes are the features. And we want to find the classes, so the cancer types, from the gene expression. So our dataset is a bit imbalanced. So we have classes with 45 examples and other classes with 1,000 examples. But we will do as if it was not so much unbalanced. So we train a logistic regression and its accuracy is quite high. So it's about 95%. So I also showed the confusion matrix because our classes are imbalanced. And we see that we can recognize many classes without being too bad. So for a few classes, it's harder, but let's say that so the predictions are good enough so that we can try to explain the decision of the model. So, and then for the explanation, so we use integrated gradients. And this is just a slide to explain a little bit more what it is doing. It's really it's really simple slide. So we have a neural network. So in our case, it's not even a neural network, but a logistic regression, F, which goes from the gene space to the prediction space. We have samples, which contain the values of n genes. And we also have a baseline. And integrated gradient is computing the attributions of the sample with respect to a baseline. So it outputs a vector with n coefficients, and each coefficient is the attribution score given to one gene with respect to the baseline. So the baseline is supposed to be something neutral, such as a vector full of 0. And there is one property which is important, which is that the output, so the prediction of your sample minus the prediction of the baseline is equal to the sum of the attributions. So here is a little example to have more intuition on what integrated gradient is doing. So my example is with linear regression. So let's say you have a linear regression with 3 input features. So y is your prediction. And you have 3 parameters, alpha, 1, 2, and 3. So if you take a baseline full of 0, the output of integrated gradient would be the multiplication of the parameters with the input. So for us, it's a bit different because we are using a logistic regression, but the idea is the same. So what can we explain with integrated gradient on our logistic regression trained on our data? So what most papers do, so a lot of papers are doing that in biology. And what they do is that they want to see, for instance, what are the most important genes for a class. So they compute the attribution scores for all samples of a class, and then they average the genes values over all samples. And they rank the averages, and they obtain this kind of graph. So we could say, okay, these are my most important genes. So I will focus on these genes to understand what is happening. And so we did a few experiments to see if this conclusion was true or not. So first, we use our logistic regression, which was already trained. So we just used it to predict some things. And we decided to only keep in our samples a few features. And we put the other features to 0. So here on the x-axis, you can see that you can see the number of features we kept, and we keep. So each point is showing you how. So in each point we kept. So for the first point we kept one. So we kept the best features for all class. So we have 33 features kept. For the second point we kept 2 features per class, and etc. until 50. And so we can see that with only the best features, the accuracy of the model on the test set is quite low. And then with more samples, so let's say with around 500 samples, the accuracy start to reach a plateau. So we could say, okay, maybe we need to have a look at the 500 best genes and remove the other ones. So I also plotted two curves, which are the precision and the recall. So so the precision is saying if I have one example classified as the Kirk class, so it's a cancer type. So if I have one example classified as a Kirk cancer, is it really a Kirk cancer? And the recall is saying if I have a Kirk sample, am I classifying it correctly? And with these both curves, we see the same trend. So let's say, okay, we should keep 500 features for each class and study at them. But indeed, we did another experiment, which is the contrary. So instead of keeping the best features, we removed them. So here we have all features. Here we removed 10 features, so the 10 most important features per class, etc., etc. And we see that the performance decrease only when we have removed 10,000 features. So it means that if we remove the 10,000 most important genes, we are still good in our prediction. So indeed, it only shows one thing, which is that our data are highly redundant. So we already have the intuition of that because the classification was really easy. But if we were only looking at the average values of the integrity gradient, we were missing this fact. So it's important to do this kind of check. So this is the same, a similar experiment as the first one, where we keep random features instead of here the best features. And we see that with random features, we are lower than with the best features. So the best features are maybe a little bit more important than the other one, but they are not the only one which are important. So this is for the first question. So the second question that interests us is, do the most influential genes differ between classes? So because we have, for each class, we have important genes, let's say, and do these genes are different between classes? So they are supposed to be, but we just wanted to check it. So we took the attribution scores and we computed a t-SNE on it, and we show our sample. So here there are only 3 classes. Each point is a sample, and they are well classified. So the attribution should rely on different genes. So we checked that. So we did a PCA on this data, and we plot the classes in the first 2 principal components. So we see that they are well separated. So we did a toy experiment, let's say. So we just took the genes which had the highest values on the first principal components and the lowest values on the first principal component and the highest values on the second principal component. And we tried to see if these genes have high average values for the classes. So if they have, it means that the most the most important genes for the class are also the ones that enables to discriminate the classes. So I did the experiments with the BRCA class, which is this one. And so if we look at the genes with the biggest positive values on the first principal components, we see that the attribution score are really small. So this is this part of the image. If we look at the one with the biggest negative values, they are quite high. So it's this part of the image. So this is coherent. And finally, this is with the last part of the image, and it's also small. So, so we can say that the most important genes are indeed the one which enables to discriminate the classes, which is coherent. And so one last question that we're interested in is, does the average that we showed represent individual sample? So for instance, this is the ranked genes based on the average for one class. And here below, we have one sample, so the attribution score for one sample. And for this one, it's not too far from the average. So this is another sample, and it's still not too far from the average, but this one is a bit higher than the other one. And we also have this sample. And in this case, so it's completely different because there are important genes which have a negative impact on the prediction. So this highlights the fact that if we only looked at the average, we miss the variability we have within our classes. And this is something that we don't want to do when we work with biological data. So to conclude, sorry, I have it a bit long. So to conclude, here I just show you small experiments where we try to explain the prediction of a logistic regression on a gene expression data set. And we show that we are indeed highlighting the most important genes for class discriminations for from the point of view for logistic regression. But in the interpretations, we have to be really careful with the redundancy that we have within our data sets and with the variability that we need to highlight within a class. So we are working on characterizing redundancy and variability with clustering techniques or cross-signal processing techniques. And we would like to extend that to multimix data that are available. So thank you for your attention and